Good evening, Pratham. So, last time we had discussed the clock genes, then we had discussed about the various other pathways related to the brain, right? We had discussed the thalamus, then epithalamus, then pineal gland. Okay, quite interesting discussion we had. Okay, so let me forward the link. So today we'll be discussing about the neurons and how does the brain produce electricity, okay, the action potential, and also we'll be discussing about a little bit more about the clock genes itself. Okay, I'll tell you the some of the latest information regarding the clock genes itself. So what happens is in the last class we had talked about the clock gene and about the body's own internal clock. Good evening, Gayatri. So I'm Rahul Saha, and last time we had talked about the body's internal clock, the suprachiasmatic nucleus present in the hypothalamic system, right? And how does it control the pineal gland to make the or to cause the release of melatonin, which is then which is then produced from a specific amino acid known as tryptophan, right? So those things were discussed. But have you ever thought about the timing of body's own internal clock? Right? Does the internal clock of the brain have its own timing? So the research says that the body's own internal clock, the brain's own internal clock, runs 31 minutes, you can say more, or runs for 30 minutes, 31 minutes more than the time required for the earth to complete its spin, meaning that the internal clock of the brain is 24.5 to 25 hours long. Okay, so we have our internal clock. We are not dependent on photons always. Okay, so we have our internal clock, but the thing is, it runs 24.5 to 25 hours. It's that long. So the thing is now the intricacy increases. Now what happens is the entirety of the human brain, okay, the clock becomes 25 or 24.5 hours long. So when it is that long, what happens is we need to be, okay, on some kind of control in order to adjust the body's clock, because the day is of only 24 hours. If it is not adjusted, okay, then the entire clock, the sync of the internal clock in the brain and the 24-hour day cycle, that sync will go haywire. Okay, that sync will not stay correct. In order for that sync to happen, then we have the connection of photons, then stimulating the SCN, okay, input of, you can say, sunlight through the eyes, in order to reset itself each day. That means the brain, the internal clock of the brain needs to reset itself each day. Okay, so when humans are allowed to run off their body's clock apart from input from the sun, okay, by being kept in continuous darkness, what could happen is the body's daily cycle needs to lengthen to about almost 25 hours then. Okay, and when people or animals lack the genes that help control the clock cycle, okay, the sleep wake cycles can stay, or you can say can stray even further, because we have clock genes. So our sleep wake cycle is of 25 hours long, but say in some animals those genes may not be present. Okay, the clock gene, the BMAL gene, all those things may not be present, the period gene, so they can even stray, you can say, further and further. So that's what I was willing to convey to you today. And one more thing is that we have something known as epigenetic clocking also. Epigenetic clocking is there. And have you felt, this is a very genuine question, that time flies by too soon when you are happy. Have you felt like that? Like sometimes the thing is everything is going so great. The time flies by too soon when everything goes great. Right? We say that good times end early. Why is that? Because there is a specific neurotransmitter known as dopamine. And due to that specific neurotransmitter, what happens is that dopamine, there is a recent research where it has been unveiled that dopamine can, you can say, increase or decrease our perception of time. And we may have two sets of, you can say, clock or two clocks inside the brain. One is the SCN and entire SCN with the pineal gland. Another one is a different clock located at the striatum, okay, which is one of the parts of the basal ganglia which I was explaining to you last time. Globus pallidus, putamen, striatum, right? Lentiform nucleus. So striatum is one of the parts where I told the entire integration of the memory takes place, and the striatum is also the part where dopamine can act and can cause excessive, you can say, pleasure, increases the pleasure, and can also cause increase in the perception of time and enhancement in the time flow. It has been found in research that if the, you can say, mice or the subjects were given dopamine, okay, dopamine injection, L-dopa injection, levodopa, okay, so their sensitivity of time, it's, you can say, rapidly increased. So they could feel that, oh, it's a very great time, but they felt like when the great time got over, the time went off too fast. That was seen. And in people with Parkinson's disease, where the dopaminergic neurons they die, the dopaminergic neurons, the neurons which secrete dopamine, okay, the dopaminergic neurons, they start to die. What happens there is the dopamine is not produced too much, and again the body's clock is adversely affected. So they don't get that feeling, okay, of, you can say, the perception of time is not really observed by those specific people who are suffering from Parkinson's. Mainly in the midbrain, there is a region known as the substantia nigra. So there, the dopaminergic neurons starts, their death starts happening. They start to degenerate. Again, alpha synuclein theory is there, but that is not in your course. So that is one thing. Okay, dopamine and, you can say, perception of time. One more thing is, you can say, people at different, you can say, rates, with some individuals developing both characteristics and diseases related to aging earlier in life, you can say, than others. It keeps on happening. So understanding more about this biological age could really help us learn more about what can we do to prevent diseases, right? And associated, especially the diseases which are associated with age, such as dementia. You might have heard of dementia, that you say that people say that when we get older, okay, as the brain ages, the brain becomes lesser and lesser as per the volume, the atrophy happens, and then the neurons are lost, and then the cognitive decline starts to set in. Okay, so scientists have again found out that there is an epigenetic clock in the human brain. So as a result, you can say, of that, they have done the experiment on the human brain tissue samples. Okay, this new epigenetic clock is far more accurate than any previous version of the clock, okay, which has been found. Okay, so what happens is, you can say, the epigenetic clock is a very exciting stuff, okay, and it has the potential to help us understand the mechanisms involved even in aging. Okay, so that is the thing. So they have identified multiple DNA methylation sites, okay, which can optimally predict age in the human cortex region of the brain. Okay, so one, you can say, 347 DNA methylation regions have been found till date. So epigenetics is nothing but you change the expression of the DNA without even touching the DNA. That is known as epigenetics. Epi means on top of genetics without touching the nucleotide. Okay, suppose this is the number of nucleotides A T G C C G A T C. Suppose this is the sequence of a gene. Okay, without causing mutation means without cause changing this A to C or anything, I am able to increase its expression means more protein can be made from this gene, or stop this gene, silence this gene completely means no protein can be made. How can it be done? Via various methylation, because histones they wrap around the DNA. So I can heavily methylate the histones in a way, trimethylation at specific amino acid residues, mainly lysine and arginine are present. So that way I can completely shut down the expression of the protein, because then in class 12th, I'll be teaching you there is a specific protein known as rRNA polymerase that should come and bind to the upstream region of the gene here before the gene, and then mRNA can be produced. If this is blocked by epigenetic changes, then mRNA cannot be produced and then protein cannot be formed even. Okay, so this also happens in our human brain, epigenetic clocking. Okay, very, very important. No issues, Samesh. So I was just explaining various types of clocks in the human brain. Okay, so why do we feel the perception of time? Okay, the mice they do laugh. Pratham, they do laugh. Not like us, like they'll not really laugh in a way that we laugh. But what happens is the scientists have found out that once you tickle mice or once they are really doing their maze running and all, their dopamine level increases in the brain at that point in time. Okay, they have done a specific type of MRI known as functional MRI, functional magnetic resonance imaging, fMRI it is known as. So that technique shows you the activity of various brain regions. Okay, so once we tickle them or once we dance, not once we dance or once the mice dances, they move nicely, so they are really happy. Okay, so every creature experiences that thing. Even Drosophila experiences that thing. The fruit fly, that is one of the best research, you can say, paradigm for any scientist. Drosophila melanogaster, the fruit fly, because all the genes, clock genes which are present in human body, almost identical genes are present in Drosophila. Okay, so a lot of sleep related work has been done in Drosophila. Now you will say that does fly sleep? The answer is yes again. They also sleep. Okay, so the thing is we have extreme, you can say, similarity with the other creatures of nature. But the thing is we think that laughing means showing teeth. Doesn't mean. Laughing means you feel the sense of happiness. Okay, why dopamine? You can, everything is neurological. The rage, okay, the feeling of murder, anguish, whatever is there, it is all neurotransmitters in the end. Okay, you can measure different neurotransmitter levels and then plot a graph. You can tell what is the exact, you can say, emotion of the person at that point in time. Directly you can predict. This much science has progressed. That is why neurology is such an important aspect. You can teach a person how to control his thing, behavior, tone down or change everything based on his specific brain chemistry. And everyone's brain chemistry is different. Maybe the amount of dopamine you secrete is less than that of mine. Okay, that our DNA is somewhat similar to bananas. Yeah, that is correct. You might have seen on this Zomato app, right? When you click 60% banana. So our DNA is 99, more than 99% similar to the DNA of chimpanzees. Okay, but the point some percent of the DNA change will lead to rapid protein change. Okay, that is why our evolution, if you see, our ancestors are who? Our ancestors are viruses, bacteria. Even we have, if you have studied the cell chapter properly, so you might have studied that the mitochondria, okay, the chloroplast, so they are not our own organelles, right? They were engulfed. Okay, if you have seen, if you have learned that theory, so the thing is, they are the remnants of bacteria. Why? Because the mitochondria have circular DNA. Chloroplasts have circular DNA. Bacteria had or has circular DNA. Okay, so still again coming back to the ribosome, 70s ribosome, we have 80s ribosome. Okay, so the thing is that is a remnant of bacteria, viruses. Even in our DNA we have remnant of viruses also. Bacteria is there, now viruses, that is known as transposons. We have retrotransposons, line and sign, jumping genes. It's not in your real course, little bit is there in your, you can say, biotechnology course, just the introduction is there. So those jumping genes are there which are direct descendants of viruses. Okay, so our DNA matches with a lot of, you can say, individuals, even viruses, bacteria, banana, chimpanzee. Okay, so we have evolved from them. They are our ancestors. Okay, so coming back to the neuron theory. Okay, so basal nuclei we had covered. Okay, so I'll just tell you the function of the basal nuclei or the basal ganglia. Okay, the functions. So when we walk, okay, just listen to this. When we walk, the basal nuclei, it controls the cycles of arm and thigh movements that occur between the time you decide to start walking and the time you give the stop order. Just think how amazing it is. That is the work of the basal nuclei, okay, which is present deep within each cerebral hemisphere. And as you begin a voluntary movement, what happens is the basal nuclei will control and adjust the muscle tone also along with the cerebellum. The basal nuclei also controls and adjusts the muscle tone, particularly in the appendicular muscles. This one, not the skeleton, appendicular, hands, legs, appendicular skeleton, appendicular muscles, axial skeleton, axial muscle. Okay, especially in the appendicular muscles to set the body position correct. So when you decide to pick up a pencil, suppose I'm using a digital pen here. So suppose I am deciding to pick up this specific pen. What happens is I am consciously reaching and grasping with my forearm. Suppose this pen is kept here. I'm consciously, because I have to write, so I'm consciously reaching and grasping it with my forearm. Okay, wrist and hand, while the basal nuclei operate at the subconscious level, to position the shoulder or my shoulder and stabilize my arm, because I am going to, you can say, reach out there, but if my shoulder is unstable, will I be able to reach the pen? I can never reach the pen, right? So I can never ever reach the pen if my shoulder is unstable. My arms will not reach the pen. Okay, as simple as that. Okay, try it is balancing your shoulder, unstabilizing your shoulder and your arm, this entire thing, and then try to reach, okay, with your wrist and hand or the palm of your hand. You can never do so. Okay, so that is again done by the basal nuclei. Okay, that I am trying to reach, suppose, but this is all vary. This is not stable. This is moving like this. So can I reach the pen? No. So who is controlling it? You are not doing anything to control your arm. Are you doing it? You are just extending your hand like this and grabbing the pen, grasping the pen. But subconsciously, this is very, very stable. You feel this is stable. You feel this is normal. But the thing is, this being normal is directly controlled by the basal nuclei. If that has any problem, you have to specifically control this like this, and then you will be able to grasp, or you can say grab the pen, or grab anything, the ball or bat or anything else. Okay, so that's how tricky our brain is. Okay, so then we'll be moving on to the neurons. Okay, now we'll be moving on to the neurons. So I'll be showing you with directly. I can show you the image of the neuron. Okay. So before showing, I can give you a specific table. Okay, that would be much better for you. So the thing is, we have something known as the central nervous system. All of you know the brain, the spinal cord is known as the central nervous system. Now, what are the differentiation of the nervous system? First, let's talk about that. Then we can shift to the neuron. Okay. So central nervous system, CNS. Okay. Once we have talked about CNS, okay, then what happens is we have special sensory receptors, okay, which monitor, you can say, smell, which monitor the taste, which monitor the balance, which monitor the hearing. Okay, special sensory receptors. That is there. Okay. Then we have visceral sensory receptors, which will monitor the internal organs. Visceral means very, very internal, like parietal layer, visceral layer. Visceral means the internal organs. So if you are taking a bump in the internal organ, so it must be monitored, right? If you are taking a kick on the liver, if the liver is getting hampered, it is monitored by the brain, okay, by the sensory neurons present. Okay, so that is known as visceral sensory monitors. Okay. Then we have somatic sensory monitors, which will directly know what is happening at my joint, my muscles, and my skin. Okay, that is the somatic sensory monitor. So from all these three, they integrate together. Okay, they integrate together, and the sensory neurons, okay, will then travel from the peripheral nervous system, okay, from the PNS to the CNS. This is known as afferent neuron. Afferent means going towards the brain, that is the sensory neurons, taking information to the brain. So paralysis means, okay, sometimes you have something known as what your the great scientist Sir Stephen Hawking had. He had something known as amyotrophic lateral sclerosis. In that disease, the sensory system is working all fine. What happens is his motor neurons degenerated. Okay, in ALS, the motor neurons degenerate or die. So he could feel everything, but could not move his hands. Okay, he could hear you if you touched him, he could feel your touch, but he himself would not be able to move a muscle. His motor neurons, they got degenerated. But in paralysis, what happens is motor sensory both may get degenerated. When somebody touches you, you may not be able to feel that as well. So the sensory neurons, they have also degenerated, and you cannot move your hand, means the motor neurons have also degenerated. So there is difference between sensory and motor. Okay, and suppose a person cannot move his limbs, you have to ask that person by touching him whether are you able to feel the touch or not. And if he is able to feel the touch, means his sensory neurons are still viable, are still working properly, appropriately. His only motor neurons have degenerated. Okay, this is known as afferent neuron or sensory neuron. Then the motor commands or the efferent one will come down into the PNS. Okay, slash efferent neurons will come down into the PNS, okay, and produce somatic nervous system and autonomic nervous system. Okay, somatic and autonomic. Always know. Okay. Somatic, that will control the skeletal muscle. Another is autonomic. Autonomic has two steps: parasympathetic. Okay. Another one is sympathetic. Autonomic means the nervous system or the nerves or the neurons which control the organs that is beyond the control of your conscious level. Like, can you control your heartbeat? Can you control the peristalsis that is happening in your large intestine right now? Okay. Can you control the kidney filtration process? Can you control the vasodilation and vasoconstriction happening in the artery in the tunica media or the muscular region of the artery or the arterioles? No, that is all autonomic. But I can move my hand, the skeletal muscle. I can move my bicep, tricep, my you can say pectoralis major, minor, trapezius. I can do all that. Okay, that is all skeletal muscle system. So I can move that. Conscious control, voluntary muscle, skeletal muscle. Then smooth muscles and cardiac muscles. They are involuntary in nature, right? They are not in our conscious control. So the thing is, they are autonomic in nature. Okay, so that is efferent motor means again their brain is controlling them, but autonomically. Okay, automatically you can say. Now you will ask a question: why everything is not, you can say, sympathetic? Sorry, everything is not somatic. Just think from the evolution perspective. If you had to control your heart rate also, your kidney filtration also, this, that, so would you be able to concentrate on the work? Wouldn't your productivity be hampered? Right? So evolution again took a detour. It analyzed the natural selection and acquired variation. As per the variation, it produced the autonomic nervous system and gave most of the control to the autonomic nervous system so that we can concentrate on the work at our hand and can increase our productivity, right? So that is again due to evolution only that we have this somatic plus autonomic nervous system. Otherwise we will be completely jumbled up, puzzled up by what to control in our body, because there are trillions of capillaries, uncountable. Which capillary will you control? Okay, you will go completely berserk, you can say. So that is how this entire brain setup is controlled, or the setup exists. Okay, so smooth muscle, cardiac muscle, the glands, the adipose tissue, they are all controlled by again the parasympathetic and the sympathetic branches of the autonomic nervous system. Now, parasympathetic, I'll just give you an example. We'll talk about this in detail in later classes. So, suppose you have gone for a strenuous activity, okay? That time you will be thinking, or you might be running, okay, in your games period. Like now you don't have, but suppose you are gymming, or you can just analyze what you used to do in your previous games periods when your school was open before the pandemic. So we run a lot, right? During that time, the heart rate increases. The heart rate is bound to increase because the muscle oxygen demand it increases, right? So you need more and more oxygen supply. So because of that, the heart rate, the inotropy of the heart means the pumping power and the heart rate itself, tachycardia occurs. So more and more oxygen is thrown onto the muscles because blood will contain hemoglobin. Each hemoglobin molecule can carry 4 oxygen molecules, and then it will be delivered, and more glycolysis happens. Sometimes aerobic respiration, depending on your conditioning. Then lactic acid metabolism, Cori cycle, and all will come into action. But you need a lot of energy. So because of that, the muscles tend to get more and more blood supply. So that time, the what happens is there is a hormone known as epinephrine and neurotransmitter also epinephrine and norepinephrine, simply known as adrenaline and noradrenaline. What they do is they bind to the heart to their own receptors known as beta adrenergic receptor GPCR type, that I'll explain it to you in later classes, and they increase the heart rate. Okay, they elevate the heart rate. Okay, elevation of the heart rate, so that the pumping action increases, the heart rate increases, and more blood is supplied to the peripheral tissues, especially the muscles, which are being used when we are running or fighting or playing whatever you are doing, cricket, basketball, football, whatever game you are indulged in. Then when you return to the classroom, that time you will be feeling that your heart is pumping fast. That is known as tachycardia. Simply you can call it palpitation also. But after a period of 5 to 10 minutes, when the teacher has started teaching, fan has started setting, if you have AC is cooling you down, then you will be feeling after a certain point in time that now you don't feel your heart rate any longer. Okay. Now the heart rate has again gone down to its normal level, 70 to 72 beats per minute. Now why is that happening? That is happening because there is a again a different neurotransmitter known as acetylcholine which has been released. Acetylcholine, ACH. Okay, this one is epinephrine. So acetylcholine has decreased the heart rate back to normal. Okay, so that is parasympathetic, inhibitory. Okay, this is sympathetic excitatory, you can say. So this is one way of describing parasympathetic and sympathetic nervous system. Okay, more about that we will be talking as we proceed in the lecture. Okay, so this is the parasympathetic and sympathetic messages. Okay, so next. Next is coming to our most important, you can say, conclusion, which is the neuron. Okay. Now, structure of a neuron. So I'll just show you an image of neuron. Okay. And describe the structure from there only. Neuron structure. Again, look at any of the images. Okay, this is good image. I'll just amplify it for you, magnify it. Okay, now look at this image. Okay, and I'll describe each thing one by one to you. Okay, just look at this image and I'll describe each thing one by one. Okay, now what happens is the cell body of the neuron is also known as soma. It contains a large round nucleus with a prominent nucleolus, okay, which we had discussed already, okay, and say parts of the nucleolus, okay. So round nucleus with a prominent nucleolus, this is the cell body or soma, okay. What happens is the cytoplasm that surrounds the nucleus in this cell body, okay, or cyton, is known as the perikaryon. The cytoplasm surrounding the nucleus is known as the perikaryon. Always remember. Neat question. I'm writing it down here. The cytoplasm surrounding the nucleus of the cell body is known as the perikaryon. Okay. Next, again coming back to the image. Now the cytoskeleton of the perikaryon. Okay, perikaryon will have the cytoskeleton, right? It will be having neurofilaments, which are the microfilaments, and neurotubules, which are the microtubules. Okay, so also neurofilaments are also very similar. Okay, to more intermediate filaments than microfilaments that has also been found in research. So you can say that neurofilaments are equivalent to intermediate filaments. Okay, I don't think you have studied intermediate filaments and all properly. So in one of the extra classes I teach you that desmin, lamin, keratin, all those things. Okay, and the neurotubules, okay, are very similar to the microtubules. Like you have alpha beta tubulin, which are the monomer, the gamma tubulin. Okay, so those things I'll teach you later on. So that is how the trafficking will go on inside the neuron. Without the cytoskeleton, the trafficking cannot go on. Okay, you cannot transport one protein from one location of the cell to the other. Suppose you want to transport something from area to the plasma membrane. Without this specific cytoskeleton, which are like the bridges, which are like the roadways in the cell. Okay, the cell's skeleton, cytoskeleton. Okay, just like the bones, which give the prominent structure to the body, the cytoskeleton gives prominent structure to the cell, provides specificities for, you can say, transportation, traversal, retrieval, all those things. So if that doesn't happen properly, the cell will suffer and may die also, because everywhere the protein needs to be replaced, because it has a half-life, it will degenerate on its own. So you need to transport proteins from RER which are being synthesized to various organelles of the cell. Okay, that is done by using microtubules, microfilaments. Okay. So also what happens is the neurofilaments, okay, they are called as neurofibrils. Okay, they are called as neurofibrils and which extend into the dendrites and axons and provide internal support to them. Because you might have seen in a neuron is the longest cell in the human body, not the largest. The largest cell is the ovum, but the longest cell is the neuron. So if this is like a thread, it may not be stable. So who will provide them the stability? That is the neurofilaments or neurofibrils, these dendrites. Who is providing them the stability? These dendrites are what? These dendrites and axon are nothing but the processes of the cyton only. Means the, you can say, extension of the cell membrane or not. The extension of the cell membrane is the dendrite, is the axon. So they are known as the processes emanating from the cyton itself. Okay. So these are the processes which emanate from the cyton itself, and they help to, or they are stabilized by neurofibrils or neurofilaments, which are equivalent to the intermediate filaments. Okay, they need to be stabilized, otherwise they lose their stability and may break off, right? So next thing is, the perikaryon also contains the organelles. We all know that, okay, that provide energy and synthesize organic materials. Means the neurotransmitters also. Means it has got numerous mitochondria, okay. It has also got free and fixed ribosomes, okay. The rough endoplasmic reticulum, RER, okay. Then Golgi apparatus. So all those organelles are present, okay. Lysosomes, peroxisomes, all are present in a neuron, okay. And mitochondria would generate energy via oxidative phosphorylation method, which I'll be teaching you later on in the biology classes. And in biotechnology, I have completed the Krebs cycle. So after that, we may switch to the ETS or errors in cell division as per you see and you deem fit. Okay. So the ribosomes and RER, we all know that they synthesize proteins. Okay. Some of the areas of the perikaryon, that is the cytoplasm present in the cell body, they also comprise of, you can say, clusters of RER and free ribosomes. Okay, clusters of RER and free ribosomes. These regions are known as Nissl bodies, NISSL, Nissl bodies. These regions, they stain darkly with a specific stain known as cressyl violet. Okay, cressyl violet. Again, a neat question. Cressyl violet, this is a specific stain. Okay. Stains darkly to the cluster of RER and especially RER. Okay, they are known as Nissl bodies. Okay, and why are they called as Nissl bodies? Because they have been named by after a German scientist, in order to pay tribute to that German neurologist Franz Nissl, who was the first one to describe them, to discover them. Okay, now Nissl bodies are stained so dark that they give a gray color to the areas containing them in the neuronal cell bodies. Okay, so that is why the term gray matter has been associated with the neuronal cell body, okay, dendrite, and anywhere where myelination is not present, synaptic end bulb, okay, because they appear mostly gray under the microscope because of their specific staining, okay, the result of their staining. That's why I had told you that gray matter and white matter. So white matter is due to myelination, gray matter is due to mostly the Nissl body's presence, okay. So now if we come down even further, okay, if you see this, you will be having, you can say, the entire region here. Nissl bodies has not been shown here, okay, there will be Nissl bodies here, cluster of RER, okay. So now we come to dendrite, axon, and one more thing shown in the figure is telodendrian. So variable number of, you can say, slender sensitive processes known as extensions. They are known as dendrites. They extend and branch out from the cyton or cell body, right, as shown in this figure, right. They branch out from the cyton or cell body. These are the dendrites. Now what happens is, typical dendrites are heavily and highly branched. Just keep that in mind. and some branches are studded with fine 0.5 to 1 micrometer long projections known as dendritic spines. Just look here. These are the dendritic spines. This is a dendrite. Again, they will have projections, you can say. Those dendrites have further projections 0.5 to 1 micrometer in length. These projections are these ones. Okay, these ones they are known as dendritic spines. Okay, very this is a dendrite, and very significant stuff is this one and this one, the branches of the dendrite, you can say, the branching pattern of the dendrite giving rise to, or various processes are emanating from the dendrite itself after branching process, that is known as, or those are known as dendritic spines. Okay, so then what we have is, after the dendritic spines, so in the dendritic spines are given, or they emanate, why? Again question comes in. Why do we have dendritic spines and we have dendrites? Because dendritic spines will do what? They'll increase the surface area. Okay, and they'll increase the synaptic connections. The each and every synapse, it will increase the number of synapses, number of integration. That is again a thing of profit, because more synapse, more integration, more recalling power, more control over the bodily functions. Okay, that is the main role of synapse. Otherwise we could have connected the two neurons like this together. Why synapse present? Because multiple neurons can integrate, can come and can integrate, can relate to, can connect with one neuron. Okay, thereby one neuron, if it has a lot of dendritic spines, it can connect with various synaptic end bulbs of the presynaptic neuron or the neuron preceding itself. This is the postsynaptic, this is the presynaptic, they are being connected. So that is why it can, or it enables the postsynaptic neuron to attach to various presynaptic neurons and receive the neurotransmitters, and thus cause extensive interrelation and correlation and integration of nerve impulses. Okay, now in the CNS, a neuron receives information from other neurons primarily at the dendritic spines only. That is the message I wanted to convey to you. Okay, which may represent 80 to 90% of the total neuron's surface area. Very important. 80 to 90% of the total neuron surface area. Write it down. Dendritic spines of the total neuron surface area. Okay. Next, what happens is, now we come to the axon. Now, an axon is a long cytoplasmic process. Okay, it can be as long as a meter in length. Okay, why is the axon present? Because it is capable of propagating an electrical impulse, okay, which is known as the action potential that I have been always talking about. I'll tell you how it is generated also. Okay, there will be no stones unturned. Everything will be explained. So that is the axon. The axoplasm is also present in the axon. That is known as the cytoplasm of the axon. Just like we had the sarcoplasm in muscle cell, we have axoplasm here. The axoplasm again contains the neurofibrils. I told you, the neurotubules also, small vesicles also, lysosomes also, mitochondria also, and it contains various other organelles too. Okay, various other enzymes. Okay, but the axoplasm does not contain, very critically remember this, it does not contain the ribosome. So proteins cannot be synthesized in an axon. Always know this fact. Mitochondria is present, but proteins cannot be synthesized in axon. Okay, that's why what we need is, we need the trafficking system via the neurotubules. Okay, so the thing is, there is something known as anterograde transport, means the proteins are being carried from cyton or cell body. Okay, that is just the direction. Anterograde means forward direction, cyton to axon. Okay, via the neurotubules, okay, via specific motor proteins, okay, and there is retrograde transport also. Retro means back, right? Retrograde fashion shows you might have attended, right? Retro fashion I'm saying, not retrograde. So retrograde transport means the proteins which have got deteriorated, they need to be recycled back, they need to be removed. So the deteriorated proteins, they are brought from axon back to the cyton, okay, for recycling and reuse. Okay, that is known as again retrograde transport. You are coming back, retrograde, going forward, anterograde. Okay, and that is done by the microtubules here. There are specific motor proteins, kinesin and tinnin, but that I'll explain in the extra class. I'll give you the specificities of the cytoskeleton, okay, so that you can understand more topics much more clearly. The cell chapter also. So if you know cytoskeleton, you can understand various topics with more, you can say, clarity. Okay, so this is what axons are up to. Okay, now the axolemma is the plasma membrane of the axon. This surrounds the axoplasm. Okay, in this CNS, the axolemma may be exposed to the interstitial fluid. Okay, or may be covered by multiple processes of the neuroglia. Neuroglia I'll explain to you. Okay, neuroglia are the, just for the names that you can know that neuroglia are present 2 to 3 times more in number than the neurons itself, and neuroglia, they do not conduct any action potential, but they help to nourish the neurons. Okay, I'll tell you the explain the different types of neuroglia both in CNS and PNS. Just hold on a minute. That is the function of neuroglia. So neuroglia can also attach with the axon, okay, or attach to the axon via their cellular processes, via their extensions. Okay. Now what happens is the base or initial segment of the axon in a typical neuron joins the cell body at a thickened region, at a very thickened region, you can see here, known as the axon hillock. Okay, so here the axon joins the cell body at a very thickened region known as the axon hillock. Okay, and axon may branch also. Okay, from the or along its length, producing side branches also. Okay, it can branch from here also, producing side branches like this, which is not shown in this figure. Been shown in this figure, it is known as the collaterals. Okay, and from where the current or starts to be produced in an axon, okay, it is known as the initial segment. It is present here. Okay, the initial segment is present over here. You can see just after the axon hillock, there is a place in the axon known as initial segment here. Okay, from that place in the axon, the current starts to be produced, or the current initiates. Okay, so that is the thing. And the main axon trunk and any collaterals of the axon, they end in a series of fine extensions. Okay, those are known as telodendria. Telo means end. Okay, dendron means tree. So the end of the trunk of the axon is the telodendria. Okay, telodendria can also be called as the axon terminals or synaptic end bulbs, terminals, various names are there. Okay, so this is the overview of the neuron, and the axon then could be myelinated or non-myelinated, okay, which we will be discussing in further detail in the next class, because all the axons are not myelinated. There are 3 types of axons. Okay, in neuron, axons are known as fibers because they look like threads, so they are known as fibers. So there are type A, type B, type C. Okay, depending upon their diameter, okay, and myelination. So the thing is, type C neurons, or type C, the neurons that contain type C fibers or axons, are the neurons which are unmyelinated. So never believe that all the neurons in the body are myelinated. Okay. So type C are unmyelinated, type A and B are myelinated neurons. Okay. So that is how we proceed with the neuron and its entire structure. Okay. So the movement of materials between the cell body and the axon terminal. Okay. Again, which I was telling you about the anterograde and retrograde transport, that is known as axonal or axoplasmic transport. Okay. So I have told you anterograde and retrograde. For that specific neat thing, you need to know about some kind of terminology. So the thing is that entire transport is known as axonal or exoplasmic transport. Okay. So this is all about the specificities of the neuron, and about the transport I'll tell you that the transport mechanism, okay, is quite slow, or sometimes it is known as a slow stream. But some vesicles, they move much more rapidly, or you can say fast stream, at a rate as high as 1000 mm per day. Okay, 1000 mm per day, because one vesicle started from the, you can say, cyton, keeping the protein inside it, and they can travel 1000 mm in a day. If the neuron is so long, if the axon is very long, they can travel 1000 mm per day. That is a very, very fast transport. But usually what happens is the vesicles move at an average of 300 mm per day. Okay. So they move at a speed of, suppose from here the proteins have been produced and here the protein is needed. So suppose the distance is 1000 meter from this region to this region. So in a day it can move. That is very fast transport. But mostly it can move till 300 mm per day from here to here, the vesicle which may contain the protein. Okay. So that is how you can say fast stream and slow stream movement of the vesicles on the neuron that carry the proteins from the cyton to the axon, or vice versa. Okay, because they keep on needing proteins and they do not have their own ribosomes, so they are dependent on cyton for their protein requirements. Okay. So is there any question? Anyone? I hope everything is clear, and we'll proceed with the generation of action potential in the next class. Okay. So I don't think you will be having any queries. If you do have, then you can contact me on my personal number. I will be replying to you as soon as possible. Okay then. So thanks a lot, and let's meet in the next class. You can tell me if you have any query. Any query if you have. Okay, 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 Pratham. Okay then. Thanks a lot. Let's meet.